For everyone with an interest in NASH or more broadly fatty liver disease, Surf's Up, Season 4, Episode 32 of Surfing the NASH Tsunami. Our continued discussion of drug development and drug development presentations at the EASL Congress and the ADA Scientific Sessions starts now.
This week on Surfing the NASH Tsunami.
The paucity of data we're collecting in those phase 3 trials now with outcomes linked down the road will help us to define these type of measures that really haven't been looked at before.
We never worried about liver volume reduction in liver disease before, but the fact is it's present, it's measurable, and now we need to understand why it's occurring and we need to understand what its impact is.
I don't think that treating obesity by itself is curing the fibrogenic burden in the liver. While it might add to the defattening effect, I still think we need more liver-directed drugs.
Quite frankly, we have been dealing with an increasing burden of NASH, meaning an increasing number of people progressing to cirrhosis that are presenting for the first time to the ER in a decompensated state. So yeah, this is a huge unmet medical need.
The only way to stop people from getting to a decompensated state is figuring out who they are first and then getting them drug.
We're not going to be serious about screening until we have a drug approved.
Over the last weekend in June, the EASL Congress in Vienna, Austria, and the American Diabetes Association meeting in San Diego, California, yielded exciting new information and data on major advances in drug development, non-invasive testing for steatotic liver disease, specific implementation of clinical care pathways, and major advances in AI-assisted histopathology. Throughout July, Surfing the NASH Tsunami will share insights and perspectives on all these advances. Our month of insights continues today, as hepatology researcher, key opinion leader and Surfing the NASH Tsunami co-founder Stephen Harrison joins our regular co-hosts, hepatology researcher and key opinion leader Jarn Schottenberg, and pricing and forecasting guru Roger Green, to discuss some of the major drug development stories of last month, today, on Surfing the NASH Tsunami.
It should be noted that if you like that introduction, you should probably cherish it, because we are looking now at exactly how we rebrand this podcast, and that specific phrase with NASH and fatty liver in the middle of it will probably go away sometime during the month of August, if not the month of July. But we're here today, and I have with me only two other colleagues, but two colleagues capable of filling hours if they had them available with this topic. Jarn is with us in the 96 degree, or for those of you on the continent, 34, 35 degree heat in Mainz. Jarn, you look hot.
Hi, Roger. Well, it's cooling down towards the evening. I made it through the day. As you know, most places don't have air conditioning here, so it can be challenging, particularly for patients too, but we manage.
And we have Stephen with us. And Stephen is, 42 in San Antonio right now, or 106, depending on how you want to read it?
Well, it was yesterday. Today, it's still early yet. It's only 1 o'clock here, so we're still heating up. We're probably about the same temperature as Mainz right now, but we're going up and he's going down.
But you have air conditioning, at least somewhat.
Oh, do we have air conditioning? Yes. We have backups for backups.
Which you have to, because after the first grid blows out, you need a second grid and your own solar and everything else.
It's almost like we all have extra Freon just lying around just in case.
I like it. Planning for the end of times, you need Freon just to survive.
Or for the grid to fail again.
A distinct possibility, yeah. So at any rate, and I'm back here in Pennsylvania, where the temperature today is a balmy 32 or 33, but we've had storms and flooding. For those of you who haven't seen the news, the lower Hudson Valley in New York, which is one of the river areas near and dear to Delaware River's hearts, flooded enough that there were literally cars swimming on the streets of West Point yesterday. But it's better today, and here we're hoping. So we're just going to talk about drug development today. We talked a little bit with Mazen and then with Jarn last week, and now we've got, as Mazen calls him, the Duke of Drugs with us. So we'll just kind of dive right in. But first, icebreaker, if you will, given where we started. One thing not about the weather, good thing that happened in the last week, personal or professional. You're both brave, so whoever wants to go first, just go ahead.
Well, I guess I'll take the lull to jump in and say I'm getting older, or we're empty nesters, and my daughter has sprouted her wings and decided to do a semester at Oxford University, where I'm fortunate enough to be on staff. And so I had the good fortune to drop her off there last week, and she seems to be having a terrific time one weekend. So it's it's great to see your kids do well. It's great to see them excited about their future and what's going on in their lives. So just thankful that I still have her, and hopefully she won't get married for quite some time.
Jorn, top that.
Well, I can claim that my daughter won't get married for quite some time, so I have additional years on that, and I'll call you, Stephen, once I get there and ask about how to dampen the pain or the joy or whatever it will be. I don't know. I visited a tennis tournament with her the other day. Her team was playing well. They won. She had 2 singles that she lost against a boy her age. First one was real close, and she played good tennis. She has a strong backhand, I found out. The second one, she didn't really want to follow through, so it was fascinating to see her want to win and fight and then kind of come over a loss and have the team win. So that was, I think, an important experience, and also something I hope I can build with her further.
That's great. I got to watch my granddaughter have her first experience with backyard fireworks on the 4th of July on Tuesday. And the nice thing about backyard fireworks is they're not particularly dangerous, but they are very, very bright and noisy. And watching her eyes light up, and it was unnoisy enough that their dog was not freaked out. And it was just a great, things you get to see only once in a life, which is the first time. She totally lit up. We're taking her to Disney World in November, where she will get to see fireworks over the kingdom, and I will be with her for that. And I got to believe that's going to be an amazing experience. And then she gets breakfast with Mickey to boot. But you all will hear about that when we get to November. So that was mine. With that, why don't we dive in? Stephen, even before the conference, the thing that people on this podcast wanted to talk about was Maestro Nash, and what it did say and what it didn't say and what it portends going forward. And as the lead investigator and the guy who presented it, I think you are the best positioned person in the world outside the company, which means who can actually talk about it a little bit, to talk about it. So why don't we just start there?
Yeah, sure. Well, Maestro Nash is a continuum of a series of phase 3 trials that resmetirom has been put through as part of their package to present to the FDA. As you know, there's Maestro NAFLD 1, which had about 1,200 patients in it. It had a 100 milligram open label arm. It had a 100 milligram randomized arm, an 80 milligram arm, and a placebo arm. Plus, it had a cirrhotic arm that it wound up putting about 180 patients in that. Treatment was for 1 year. It's all non-invasive. And then they opened up a second phase 3 trial, which was an open label extension of that trial. And that's called the MAD-18 trial or open label extension trial. So with both of those trials, you're getting a large number of people who have a minimum of 2 years of follow-up. And then you get to Maestro Nash, which is the registration trial. It's the trial that gets you accelerated approval. Had to have a biopsy to get in, showing Nash with an NAS of 4 or more with really F2 or F3 fibrosis. Some F1Bs were allowed to get into the trial. And for the listeners that may not know, F1B and F2 are really very, very close to each other. And in fact, some of the work we've done have shown that demographic-wise and clinically-wise, there's no difference between a 1B and a 2. And then treatment was for 1 year, 52 weeks, at least for the conditional approval. Liver biopsy was done at 52 weeks. But the trial continues out to 54 months because we have to collect outcomes data on those patients. And one of the important outcomes is progression to cirrhosis. So another liver biopsy is scheduled for 4 years after the 52-week biopsy. So the primary endpoint was either fibrosis improvement without worsening of NAS or NASH resolution without worsening of fibrosis. And there was a key secondary endpoint. And I say that because you take a statistical hit when you add in a key secondary endpoint. But it was important to do that with resmetirom because this drug has significant atherogenic lipid reduction potential. And so LDL reduction at 24 weeks, not 52 weeks, was built into the protocol. And of course, based on the information we have from the Intercept AdCom and the CRL that was recently issued to Intercept, we understand the FDA has a big interest in safety. And so data was also presented relative to safety. So that's the Maestro Nash program. I'll jump into that in just a second. Maestro Outcomes is the 4th phase 3 trial that's being done in the drug development strategy for Madrigal to get resmetirom approved. And that is a well-compensated F4 trial that is currently underway and set to enroll between 700 and 800 patients. The primary outcome is a progression to a decompensating event.
Thank you for the overview. Let me just jump on the Maestro Nash that you presented at EASL and discuss the endpoints very briefly because there's been some discussions around the validity of liver biopsies, variability, and so on. If I remember correctly, the data that you presented, histology was read by 2 experts. They had consensus. There's AI assessment, which backs the data. And in the end, it's a conditional or it's a predefined endpoint. And we're looking at outcomes, as you said, quality of life. So the number that you show for improvement, both in fibrosis and NASH resolution, might not be equal to the effect size that a patient actually benefits, because it's just a surrogate we're looking at. And I thought that comes to my mind when I think about these endpoints, because sometimes people say, the number, it's only so much above placebo, but it might not be the benefit that the patient actually experiences. Is there something that resonates with you?
Absolutely. One of the things I struggle with in drug development, and particularly for NASH, is we get hung up on this notion of treatment effect delta. How well does the drug work compared to placebo? And you have to remember that this is an ITT, so it's intent to treat. That means anybody that signs consent essentially and gets a dose of drug is considered in the trial, which means if for whatever reason they drop out of the trial or don't have a follow-on liver biopsy, that's considered a drug failure. And so all those were taken into account when we discussed the primary endpoint. So just to jump to that, for NASH resolution, there was a 20% treatment effect delta with a 100 milligram dose versus placebo. It was 30% versus 10%. And there was a nice dose response. The mid dose, the 80 milligram dose, had a 16% treatment effect delta. And when you looked at a one-stage improvement of fibrosis without worsening of NAS, it was a 12% treatment effect delta for the high dose and a 10% treatment effect delta for low dose. And to your point, those numbers seem modest. But when you actually look at the people that had a biopsy at the beginning and actually followed through and had a biopsy at the end, this is what so-called MITT, and you look at the response rate, it's actually 39% for high dose and 11% for placebo. So, there you're talking much larger numbers of patients actually having a response. And even the low dose, 32% versus 11%, so a 21% treatment effect delta. And then when you look at greater than or equal to one-stage improvement of fibrosis without worsening of NAS, it's 33.5% versus 16%. It's almost a doubling, more than a doubling of patients actually having a response there. And then when you look at either or, like NASH resolution or fibrosis improvement, it's 50% for the high dose versus 19% for placebo. So that's just looking at ordinal scoring. And I know one of the concepts you and I both are big on is AI digital pathology and really trying to unpack what's happening in liver biopsies that may or may not be visually obtainable to the pathologist on ordinal scoring. And it's important to realize on ordinal scoring, based on the NASH CRN reading, pathologists really don't focus on central vein relative to what's happening on ordinal score reads. And when we look at some of the data that was presented as a late breaker, so this was Maestro NASH data looking at histoindex or Q fibrosis, the treatment effect deltas were even higher than what I just told you. And if you account for steatosis regression and liver volume regression, it's even more pronounced. And you can begin to unpack where the effect on fibrosis is with resmetirom. And it turns out it's perisinusoidal, perivenular, and central vein, more so than portal tract or chicken wire. And so that's interesting because that's in an area that's not commonly read out by pathologists, particularly the central vein area. So anyway, I don't know if that's worthy of a comment back from you or if you want to infer anything from what I just said, but it just adds to what you're saying.
My only comment would be that I am aligned with you, that I think the way we use the 5-tier classification system of fibrosis in NASH does not capture all the beneficial changes we're seeing in patients, even outside of liver histology. And you've mentioned the secondaries with LDL improvement. And also the very interesting fact, and we're just understand how resolution is happening in the liver nodule and which type of fibers are actually de-resolving. So that will be moving forward. Very interesting. And it's the same for me. I think there's much more patient benefit than just the number that are currently reported. So we'll find out, I think, with the extension studies, as you've mentioned.
Yeah. And I think the liver liver volume reduction piece is one that, I sit here and I think, okay, what can my contribution to this field be? Drug development, I get that. I think another area besides AI digital path that I really want to focus on for the rest of this year is what's really going on with liver volume reduction. Because if you look at the resmetirom data, you're getting about a 25% reduction in liver volume. Now, what other trials have looked at liver volume? Mozart did, 89 Bio did, Akero did, and resmetirom did. So you have two FGF21s. You have Mozart, which is a very, very early study that Rohit Loomba worked on, and then the resmetirom data. So let's just and then pemvidutide, which is a GLP-1 glucagon agonist that I presented updated data on at EASL. If you look at PEMV, the liver fat content reduction is better than what you get with resmetirom. But the liver volume reduction is around 20%, whereas with resmetirom, it's around 25%. So here you're talking about a 50% liver fat content reduction with resmetirom, about an 80% liver fat content reduction with PEMV, but you have an enhanced liver volume reduction with resmetirom. And this is the quick conclusion is this isn't just fat related. It's not just improvement of liver fat content. There's something else going on that I believe is going to have an impact on how a patient feels, functions, or survives. There's some data that's been published in Liver International. We talked about that journal earlier before we started on the podcast. In that journal, it shows that if you look at overall mortality, the size of the liver is more impactful than the FIB4. So it makes sense that if we're improving liver volume, that could mean something positive to an outcome situation. So what is it? Between now and AASLD, I am working diligently to try to understand this better. How much of it is water weight relative to inflammation? How much of it could be glycogen? Is there some sort of vascular impact, vascular flow impact that could be meaningful? We know from the cirrhotic trial with resmetirom and in the MAD14 study that liver volumes, two things. Number one, in cirrhosis, liver volume in NASH is higher than in a healthy person. That's completely disparate to what we know in viral hepatitis. When you develop cirrhosis in viral hepatitis, you get a very shrunken nodular liver. In the setting of NASH cirrhosis, you have large livers, and we know that. A normal liver is about 1,500, 1,600 cc's. Cirrhosis, at least in the resmetirom trial, the average was around 2,200 cc's. There was a liver fat content reduction that occurred in that trial that also was linked to spleen volume reduction and inversely associated with elevations in platelet count. There's probably some vascular issue there as well.
Hey, Stephen, can I step in 1 second and make sure I understood what you just said, the way I think about stuff, which is that the cirrhotic livers in the resmetirom trial were about 25 or 30% larger than a normal liver would be, and the reductions that resmetirom generated were about 25% back from there. Did I get that right?
Yes. We can measure liver volume relatively easily with MRI, but we don't do a lot with it. Okay, so Jorn, you're big into the NIT space, more so than I am. And so maybe liver volume reduction could be used as a non-invasive test for fibrosis improvement. Why don't we look at liver volume content reduction of a certain percentage correlating to a one-stage improvement in fibrosis?
I think that's a great idea. And the paucity of data we're collecting in those phase 3 trials now with outcomes linked down the road will help us to define these type of measures that really haven't been looked at before. I mean, you mentioned HCV, but who was interested in liver size in HCV? Everybody looked at eliminating the virus and everything else was secondary. So I think even beyond, helping patients, the advancement of science we see through these large trials with the linked NITs and MR data that's being generated is unprecedented in this area. And it'll for sure help us to develop new concepts on how to assess disease progression, regression, and liver disease over the next years. So I'm aligned there.
So a couple of comments. First of all, you're in the episode that we did, the extrasode that we did with Arun Sanyal and David Kleiner last month. David actually talked about that whole idea that hepatitis is really easy. You get rid of the virus, then everything follows. And he wishes that NASH was that way, but it's not. You never really know what's going to happen. Things go up, things go down, things go in all different kinds of directions. And I think as a result, secondary measures that might not matter in a linear predictive world do matter, number one. Number two, part of my statistical training is that every time you go from a multidimensional world to a world with fewer dimensions, what you're trying to do is the best job that you can of predicting what matters. And that's when you do these in discipline multivariate kinds of ways. If you think about the history of how NASH CRN came up, not to fault it any more than anybody else does already. That's not a multivariate exercise. That's a qualitative assessment of a semi-qualitative assessment to try to build a semi-quantitative assessment. So we don't really know what matters. We know some things we can measure, but we don't really know what matters. And Stephen, this is the third time I've said this in a week on this podcast. I thought your comment on liver volume in the Maestro-NASH presentation was, to me, the single most eye-opening thing I heard in that entire session. Maybe in all the drug trials in the entire meeting, because it points once more to how little, you take that in the SEMO trials where after 72 weeks, we've got a bunch of liver fat reduction and no regression of fibrosis. And those two facts tell me how little we know.
Yeah, you're exactly right. We never worried about liver volume reduction in liver disease before. That hasn't either because we didn't need to, or because we didn't have the technology, or a combination of both, or additional factors. But the fact is, it's present, it's measurable, and now we need to understand why it's occurring, and we need to understand what its impact is. And, we struggle with understanding response to therapy. I think that's going to be one of the issues also with resmetirom once it's in the approval pathway. And we know that the NDA is currently being filed right now. We know also that there's a request for priority review, or there will be. One of the issues that's going to come up is how do we monitor response to therapy? And in resmetirom, I showed this data from the forest plots that I presented. MRI-PDFF is very predictive of response to therapy, but not everybody has MRI-PDFF. So it's hard to measure liver fat content reduction accurately outside of that. We obviously can't measure liver volume reduction outside of that, but we do have something called sex hormone binding globulin. And what I showed there was that increases of at least 120% or more were correlated very nicely to response relative to placebo. So maybe there's an opportunity there. I like ALT as well. That drops very nicely with treatment. However, not everybody with NASH that has advanced disease has an elevated ALT. So I don't think that's going to be ubiquitous across the board as a measure of response to therapy. But before we dive into that, I think we'd be remiss if we didn't spend a bit of time talking about the safety of the drug. Because when we think about therapeutic index of drugs, safety gets an equal billing to efficacy. And that's no more true than in the setting of fatty liver, where we're talking about millions and millions of patients, and our job is to first do no harm. So are we putting patients at risk by prescribing resmetirom? Well, we were looking for the F2, F3 patient, I believe, in the label. Likely is going to be the case. So do we have NITs that can inextricably tease out an F1 from an F2 or an F3 from an F4? The answer is no. We're not that great with our NITs. So what if we put an F1 on therapy? What if we put an F4 on therapy? Are they going to be injured or hurt in any way? The answer is no. I mean, if you look at the safety database that's been accumulated with this drug, thousands and thousands of patient years worth of data from F1s, F0s even, all the way to F4s. The number one adverse event is diarrhea, and nausea is number two. And both of those tended to occur immediately upon dosing. Average duration was around 2 weeks and did not tend to worsen after that point. And most patients got better. If you look at the diarrhea that's reported in the electronic data capture system, more than half of it was just worsening of already present diarrhea or intermittent loose stool. As a gastroenterologist, we struggle with defining diarrhea in clinical trials because diarrhea means something very different to Jorn and I than it does from an average coordinator just filing this in the record. So increasing stool frequency could be diarrhea. Intermittent loose stool could be diarrhea. But that's not the cholera diarrhea. There's a bit of a difference.
I agree with you, Stephen. I'm not worried about that side profile. It's the way we capture them in a clinical trial. It's very important, very rigorous, very thorough, but it's typically not what's seen in clinic or will hamper applicability in clinics.
Yeah, as a general rule. Yeah, you might have some people that have some change in the frequency and consistency of their stool. I'm sure there might be people that actually have frank diarrhea that may not tolerate it. But I think that number is going to be very, very small.
Stephen, very, very small subjective. What percentage, remind me, actually discontinued?
It was on the order of low single digits.
Yeah, to me, that's where I go, because people work very hard in drug trials to keep everybody in, despite which, as we see in some of the papers that get reported, you get discontinuation rates around GI effects that run a lot higher than that, say GLP-1s, dual agents, the retatrutide data at ADA. So when you say low single-digit discontinuation, what you're saying to me is even in the hands of a GP, this is probably easily manageable.
Yeah, this is straight from the presentation. Treatment emergent adverse events leading to study discontinuation was 2% in the 80 milligram, 6.8% in the 100 milligram, and 2.5% in placebo. And there were no study discontinuations after 52 weeks in the 100 milligram dose.
In summary, I would say it was pretty safe. And that data was exciting because it wasn't previously released in the press release. So I think this is where the novelty came in. Following up on safety and connecting this with the liver volume reduction, I think the benefit in quality of life and decrease of right upper quadrant pain is something I'd really expect from this type of MOA. And this is clearly a benefit. I've told sponsors when they ask me about, Europe and Germany in particular, you might want to show direct patient-related improvement and estimate that, because that is something the regulators over here are heavily looking at.
Yeah. The drop in atherogenic lipids, the percent change in ApoB of around 20% is something that we should not overlook. If we follow these people out, if we were looking at a MACE outcome, which we're not, that drop in ApoB has been linked in other studies to CV risk reduction. So obviously we didn't enroll high-risk CV patients in this particular trial, and that's something we're trying to move the needle on at Mosaic, our meeting that we have looking at combining endpoints for MACE and MALO, or major adverse liver outcomes. But I think the data, it's very promising. I think that it's been dosed in a large number of patients for a significant number of days, and I'm anxious to see what happens here in the next 6, 7, 8 months.
So we could stay on this one topic, something close to forever, but I'm going to try to avoid that, unless you have something else about resmetirom that either of you thinks is really pivotal to touch on. When we talked with Mazen last week, he talked about his drug development Nash's monopoly metaphor, and the idea that the two most valuable properties were the FGF21s and the GLP-1s, each of which had some really interesting issues emerge from last month, or the glucagon agonist more broadly, if you will. So we can dive into either one of those. Take your pick.
It's interesting to talk about both. To me, just reflecting back on EASL, one of the points I walked away from that meeting with was that at least at diabetic doses of GLP-1, we're not getting rid of all the fat. We're not getting rid of the inflammation and fibrosis. And these drugs that you mentioned, to include resmetirom, are additive on top of these particular GLPs dosed in the diabetic range. We also know from the FGF21 data that was not presented at EASL, but I'm very hopeful will be presented at AASLD this year, is the cohort D data from the Symmetry trial, where we actually took 31 patients that were on a baseline stable dose of a GLP. And again, most of these were diabetic doses and were randomized to placebo FGF21 or FGF21 on top of the GLP-1. And the additive incremental value was incredible when you put the two together. So I think, to me, the take-home point is the GLPs are coming to the forefront as a very active player in the field of obesity and even diabetes and ultimately CV risk reduction. I get that. Most of my patients are already on them. If they're not on them, quite frankly, I'm putting them on them. I probably prescribe more semaglutide and tirzepatide each week than I do ursodiol or some antiviral or anything else. It's just very, very common. But what I've learned is this disease, NASH, is so heterogeneous that taking a drug like a GLP that has no receptors in the liver and expecting it to deliver the mail on improving histopathology across the board in these patients is probably a bridge too far. Now, combining it with a GIP or a glucagon, the jury's still out for me on that. I mean, we have studies with pemvidutide combining a glucagon with a GLP. Obviously, there's work underway with tirzepatide. We got triple combos coming, both oral and injectable. So we'll have to wait and see. But I'm a complete believer that this class of medicine is not going to usurp the need for targeting inflammation, targeting mitochondria in the liver, targeting fibrosis in the liver, or a combination of those. And that ultimately we are still going to need combination therapy, and we're going to need multiple different choices on the shelf to choose from to manage these patients. And that's before we even talk about the side effect profiles of these injectables or even the GLP orals, which were presented at the ADA recently.
Yeah, so A, I don't know that I could agree more with everything you just said if I tried. But B, one of the things I found really telling is you take a look at the Lilly development path for tirzepatide. They're approved in obesity. They've got 4 separate indications they're listing on pipeline in phase 3, and they have NASH in phase 2. And the things they're listing in phase 3 mostly, well, one is ejection fraction, which is different. But the others are all to build a stronger case around obesity. Cardiovascular outcomes, morbidity, mortality, and obesity. And I forget what the third one is off the top of my head.
Sleep apnea, right?
Right. Thank you. None of which have anything to do with NASH. So given the size of the NASH market and the NASH population, you would think if they were confident that what they had there was a winner, that would have gone earlier. That's a GIP, not a glucagon. But go ahead, Jorn.
Yeah, the one thing, I didn't have a chance to discuss it with Stephen yet. I said that last week too. I don't think that treating obesity by itself is curing the fibrogenic burden in the liver. While it might add to the defattening effect, I still think we need more liver-directed drugs, which might be in the dual agonists with the glucagon receptors, but I think still there's room for more, and I think we're nicely aligned on this. Not always sure the investment strategies of a company or decisions are reflecting the effectiveness of the drug, but I think you really have to go into the population and do the trial if you want to find out.
So, Jorn, having done a lot of marketing research on that in my last career, what it tended to reflect was a combination of 3 things: a competitive uniqueness, probability of success, and size of opportunity. Okay, size of opportunity in NASH is so vast that you would think anything that had high probability of success with competitive uniqueness would get greenlit. In fact, one of the things we used to do was tell people change your order of indication out when you're planning for phase 2, because this is a much bigger opportunity than you realize it was, and you're more unique than you think. So I agree with you. You can't infer everything from that, but I wasn't the only person who figured this out 10 years ago. And everybody that we worked with subsequently found a way to figure it out without working with me.
But you're probably right, and I won't argue with you on that lifetime experience.
So on a commercial level, I take a look at that and I say, if these guys thought NASH, well, when we started talking about this, when I came to this space 4 to 5 years ago, people told me if you could affect a 10% reduction in body weight, you could automatically affect significant reduction in fibrosis. If that were the case, then I would think that there would be a different pathway, a different stage of clinical trials.
Automatic is a strong word. We would think that with 10% body weight reduction, you would begin to impact inflammation. That would have downstream impacts on fibrogenesis. But there are multiple other ways that activate stellate cells, right? It's not just simply a matter of losing weight. So there's more to the story here. And is the data promising? Absolutely. I mean, the data presented at ADA is landmark. I mean, if we could get an oral triple combo with a lot of our patients achieving 20% weight loss, count me in. I'll prescribe that to my patients. I think it's terrific. But one thing we don't talk about is the AE profiles. We're talking about route of administration, but we're not making the AE profile better when we're doing that. So that's still a struggle. 30% of our patients are going to not be able to tolerate these drugs for the long term, and we're going to need additional therapy. So people that want to throw the baby out with the bathwater and say, we're done, we've got an obesity drug, it's oral, it treats, it can be used by everybody for every indication, it cures everything from obesity to heart disease to kidney disease and cancer, well, then that's just a bridge too far for me right now. I do think that a lot of positive. If I could shift gears a bit in the next couple of minutes, I do think it's worth talking about the FGF21 class of drug. This drug was it got a black eye, I think, early on because of the pegvelfermin data that came out of, in the literature in 2 different publications in clinical gastroenterology and hepatology, because it wasn't effective and there was some concerns about tachyphylaxis. It's important to note that not all FGF21s are created equal, and there was distinct differences in pegbelfermin relative to what we see now with efruxifermin and pegozafermin, and even Boston Pharma's drug that doesn't have a name yet that I'm aware of, but is in phase 2B development. What we're seeing with the data coming out with Akero and the Harmony trial and with pegozafermin in the Enliven trial is that 24 weeks of therapy with repeat liver biopsy, both of these drugs are having impacts on NASH resolution and fibrosis, and even in cirrhotics. Now, the trials weren't designed for cirrhotics, but you have two different drugs studied in two different populations of patients, both with a subset that had cirrhosis on histopathology interpretation that improved between 30 and 45% of cases improved by at least one stage after 24 weeks of therapy. Now, I'm not saying that 24 weeks of therapy is enough for a cirrhotic. I'm not saying that you're going to see 30 to 45% response rates. What I'm saying is this is the first data we have ever seen with any mechanism of action where two different drugs have shown benefit in a relatively short period of time in a well-compensated F4 population. We didn't see that with simtuzumab, selonsertib, imricasan, cenicriviroc. I could go on and on. Intercept's drug just didn't show it. Regardless of where this all pans out, juxtaposing a GLP or a GLP-GIP or a GLP-glucagon GIP versus an FGF21, I think the FGF21s have the horsepower needed, albeit maybe longer treatment duration than 24 weeks, but I believe they have the horsepower needed to target a well-compensated F4 population and show fibrosis regression. I think we'll hear good data come out of Symmetry. I'm anxious to see that data from Akero, and I think we should engage with the FDA to look at regression of fibrosis in a cirrhotic population as a subpart H approvable endpoint. And I think that's a distinct difference between the whole incretin group of drugs versus the FGF21s. But you have to have balanced potency. You have to hit beta-klotho FGF1, 2, and 3. You can't just hit one as was shown with, really the pegbelfermin data.
But assuming you're right, okay, and those are the patients most in need of help, who are helpable, right? I think that's been the assumption from day one.
Well, it's the patients that are in huge need of help. It's the patients that are the quickest, that have the most urgent need of help as well. Quite frankly, we have been dealing with an increasing burden of NASH, meaning an increasing number of people progressing to cirrhosis that are presenting for the first time to the ER in a decompensated state. So yeah, this is a huge unmet medical need.
Well, it's actually two needs. One is the need to prevent people from getting to a decompensated state. And the second is to take a lot more seriously screening people before that. Because the only way to stop people from getting to a decompensated state is figuring out who they are first and then getting them drugged.
And we're not going to be serious about screening until we have a drug approved.
That is a very good comment and very important. I agree.
Right. Look, I had a conversation with somebody who's looking at NIT from an investment perspective, and they asked me, what's the fastest way to make money in NIT? And I said, anybody who could figure out how to not take F4 on CRN scale, but who could go pick off Child-Pugh or Ishak or something that could tell you who was early in the cirrhosis process, that would be hugely valuable. And to hepatologists, because everybody would know what to do if they had something to do. And it's reasonable to think we're going to have something to do.
We're definitely going to have something to do.
Yeah.
I'm very confident in that.
I just heard you say you're not going to retire for the next 10 years. That's good news for the field.
No, you didn't hear me say that.
Yeah, you haven't heard me say that I was stopping doing the podcast in 3 years when I didn't exactly say that either. So he simply wants to make sure that we all stay in place for a while.
I think the younger bucks, the Yorns, the Mazins, the Naims, they're more than capable to continue the fight.
Yeah, I believe that's right. And I do believe, Stephen, that the breadth and depth of your field of vision for having been through as many of these wars as you have creates a certain level of knowledge where long after you think you've hung them up, people are still going to be calling you.
Well, I'm just very grateful to be able to contribute what I've been able to contribute and hope I can for a bit longer, and then sit back and watch Jorn do amazing things on top of what he's already done.
Well, let me spin that idea of the compensated cirrhotics a little further, Roger. I know we're approaching the last minutes of this talk. With the success of the therapies we're going to see, I think we'll soon be discussing expansion into populations we haven't so far studied. For example, patients with HCC. So if I have a NASH cirrhotic or even non-NASH cirrhotic and I want to treat his HCC, how do I stop the underlying disease as a disease driver? Which drug, potentially resmetirom, am I going to use in an adjuvant concept to add to, let's say, chemotherapy or after TACE or something to modulate the outcome? I think the success we're building here will allow us to expand this into other patient populations and have anti-NASH drugs as adjuvants in patients that are really in need of disease modulation. That's totally speculative because we're never even thought about this yet, but I think there's good reason. If you decrease inflammation, everything, the patient might feel better.
Well, my friend Ken says all the time on the other podcast that we do that if you can achieve everything that you're thinking about, you haven't dreamed big enough, Jorn. So I love that thought. Oh, by the way, Stephen, he also says there aren't enough hepatologists prescribing enough GLP-1s. So when he listens to this episode, he will stand up and cheer for you. Not that he doesn't all the time anyway.
It's something that we face every week in practice. But then a week later, I'm faced with, well, it wasn't approved. What do you want to do now? That's the next the issue I have to deal with. So I think, Jorn, you're right. I mean, hepatocellular carcinoma is also a growing issue in this country, in your country, and around the world, largely, but not completely, due to fatty liver and it occurring outside the setting of a NASH cirrhotic phenotype. We need better tests than AFP and MRI to diagnose this at an early stage. And I think we're getting there. I mean, some of the epigenetic work being done in this field now is remarkable. And I think it's going to revolutionize the way that we diagnose HCC. But if we can treat the underlying drivers of HCC just like we can treat the underlying drivers of NASH, I think we'll see a decrease in the number of people presenting with advanced hepatocellular carcinoma, and we can really impact overall morbidity and mortality relative to the disease. That's one of the things we'll begin to unpack once we get a drug approved, and once we get a drug approved for cirrhotics. That's a huge area of unmet need that's going to be studied intensively. Maybe it's studied by investigator-initiated grants, maybe more so than pharma-sponsored, but potentially both.
Okay, so I'm going to ask for one more wild area of speculation, Stephen, and then I'm going to note that we're going to do another episode on this topic of drug development that we've learned in the last month before Jorn goes to Italy and I go to the shore. And I want to make sure that whenever we do that, even if it isn't on a Monday afternoon, that we can get you back to be part of it. The area of wild speculation is this. If 20 to 30%, if I remember the number correctly, of HCC cases are now believed to precede cirrhosis, then how is the evaluation of that likely to come forth in clinical trials, those patients likely to come forth in clinical trials, given how we now think about who we're doing trials for?
Yeah, I mean, I think we have to get better at our non-invasive tests for the disease. We can't do contrast-enhanced MRI or 4-phase CT scans on all of our clinical trial patients that are F2/3. And we can't do that serially. It's cost prohibitive. So until we develop a better test than alpha-fetoprotein or even some of the more novel tests that are out there, we need something better. Then I think it's going to be a struggle to identify these people at an earlier stage. But to your point, it's something that we need to begin to take a serious look at. We're getting after this thing one step at a time, right? The first step is let's get a drug approved for the disease. Mm-hmm. And then we can begin to unpack all the nuances of the disease. And one of those important ones is hepatocellular carcinoma.
Excellent. So let's do this. Okay, just in closing, any one thing that happened we learned on drug development in the month of June that we haven't touched on today that you think is worth putting a pin in so that when we come back to this issue, we're sure to cover it?
Well, one of the things I brought up at the meeting, and I'll leave this, I have to say it on the podcast because I like to come up with little idioms along the way. And so here's an acronym that I brought up and highlighted at the liver meeting in Vienna. And that is, we deal in a world where if you're not used to failure, you're just not going to advance science. Failure is part of what we do. It's part of Hep C. It's certainly been a part of NASH. And I have a slide I show of more than 10 drugs that have failed. And we're learning from that. So I'm going to leave you with what the acronym FAIL means. It means first attempt in learning, okay? And with that, I will stop and let Jorn have his say.
That will resonate with me. I'm trying to think of a German acronym, and it doesn't quite turn out as nice, I think, but I'll follow up with that anyway. So this is great, Stephen. I mean, the one thing that maybe the month of June hasn't talked about, there's a lot of drugs that haven't been able to, or trials and programs that haven't reported data because they're actively enrolling. They're getting closer to complete their phase 3 trial, and I think those will raise a lot of excitement. We had novel MOAs. I remember Mazen Noureddin presenting mitochondrial uncoupler at AASLD. There's always a certain area of quietness around these, but there's a lot of things in the pipeline. Now, Spotlight was on Stephen and the resmetirom data, the FGFs, so a lot of data around that, but there's more to come. And I think, also the genetic targets we've discussed briefly the other day, more targets to hit. Some failures, but for sure an attempt to learn and improve for the best of our patients. And I'm very happy to be part of that learning curve and looking forward to contribute.
So I've got two. First of all, you hear about these drug classes and then they just don't quite get there, right? Or they don't even get to the next stage. So it was nice to see the denifenstat presentation. Since I've been hearing about FASN inhibitors forever, and I'd never seen any data on them at all, and it was nice to see that this set of data pop up. I thought that was encouraging, number one. And number two, Stephen, I actually was fixing a bulky screen door this weekend, for which I used WD-40, which immediately brought me back to one of the earlier Harrisonisms, which is what would happen if they stopped at 39.
Amen.
So I don't know where we are in the WD process, but my screen door doesn't squeak anymore, and it looks like we're getting closer to some good stuff. So with that, please join us again when we do the next round of drug development, and Jorn and I will be back next week.
We're not closing the door on drug development. We're just making it less squeaky.
That's fair. That's fair. But then we also have to build a better screen so the gnats don't get through. There's a lot of work to be done here on the screen door for drug development. Totally agree. Stephen, thank you so much for joining us today. I'll be back with business report in just a moment. Jorn, I'll see you next week. Thanks, folks. Bye-bye now.
Thanks, guys.
Thank you.
Be
Great
well.
seeing you.
Stay cool.
Bye. Yeah, I'll try.
And now for the Season 4, Episode 32 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. Today, as I write and speak, NATO is preparing to admit Sweden, but not yet Ukraine. This is not a victory for Ukraine, but it does point to the scale of error and loss of power suffered by Vladimir Putin since he started this invasion in February of 2022, as does the report today that he met with Prigozhin shortly after the aborted Wagner coup attempt. Now, what speaks better for Ukraine is Joe Biden's decision to send cluster munitions to support the Ukraine side of the war. To those who see this as an escalation, do a little research into the kinds of weapons Russia has used throughout this horrible invasion. You might want to reconsider. Slava Ukraini.
It's good to have our schedule back on track.
After a month of sketchy scheduling, we're back to our regular Monday recording and Wednesday drop schedule with a great Harrison Schottenberg dialogue. Stephen will be back with other guests to be named on an episode to finish our drug development discussion that will drop on July 26th. We have 3 topics for other EASL follow-up episodes: basic science, AI, and non-invasive testing. Keep an eye out to learn which one we are covering each week and who our guests will be.
If you have ideas for the new podcast name, we want to hear them.
If you're on our mailing list or you follow us on LinkedIn, keep an eye out for an email or message soliciting your feedback on our new name. I have a favor, but we want to hear from you guys first.
We haven't discussed download counts in a while. Let's do it.
I haven't mentioned these systematically for a while. We went over 10,000 downloads a month for the second time in our history in June, and despite not publishing a single typical or classic episode this month, have averaged well over 300 per day as of Monday night, which puts us on track for 10K in the month given the quality of what's to come between here and the 31st. Also, in my mind, 25K downloads equals 1 lap, with 4 laps to every 100,000 downloads. By that standard, we cleared the 7th lap, or 175,000 downloads this past weekend. More to come.
The Vault is back this week, and it's a good one.
A year ago last week, Conversation 34.4 from Season 3 was from our first full review episode after what was then called the ILC Congress, now called EASL. It featured Stephen talking about resmetirom and Jorn discussing semaglutide. The more things change, the more the topics stay the same. So listen for how our perspectives have changed about those 2 drugs and the classes they represent and therapy in general, given everything we've all learned in the last 12 months. And with that, I'm off until next week. Thanks to Jorn and Stephen for a great conversation, to Magic Mike for producing this episode and getting Rising Tide out at the same time, and congratulations to Jake on his first full work week from the new schedule in Australia. Now all we need to do is get his fellow Oz resident Louise back on track with our schedule. We'll be back to full form and everything that works. Enjoy your weekend. We'll see you next week to discuss more news from EASL and ADA. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.